Thanks so much. And Brian, I'm so excited to see the I-SPY model moving forward because we actually use the principle that late treatment is the wrong way to study new agents. So I'm going to talk about the importance of thinking about the disease as a whole and the spectrum of disease. Now we're moving a little bit away from disease where we have mostly serious conditions to one where we have a really broad range and a broad spectrum of behavior. So I agree completely that the future of genomic medicine really depends on both ongoing discovery, but really a thoughtful application of discovery with the goal of increasing healthcare value. And by that, I mean the same results for the same or less cost. So precision medicine will often mean more for some and less for others. And it's just as important to figure out who not to treat and not to torture as it is to just add things. And clinical practice, to make these things work and to make them drive, we have to have care and research integrated so that we can have care transformed to become a dynamic engine for learning. So I don't need to tell this crowd that, two people who seem similar are not the same. You can have the phenotype of a 51-year-old with a 1-centimeter tumor, and there the commonalities go away. Depends how they're detected, it depends on the biology, depends on their genomic basis, their inherited genomic basis, what their social situations are, and their sets of values. All those things are what come into precision medicine. So clearly, we know that one size does not fit all. It doesn't fit all in treatment, and so therefore it shouldn't fit all in screening either. So I'm going to use the 70-gene classifier that actually Laura Vanfer, my close colleague and collaborator, had put forward in 2002. And the reason I think this is so important, if you think about the genes that are in every column, and you think about this, this is really a spectrum to say at the top really is the opportunity for us to do less. Because in a disease like breast cancer, especially where screening is rampant, we can discover the reservoir of more indolent disease, and we have to be very careful not to overtreat that, all the way to the opportunity to the people who are at the highest risk, who actually have very fast and progressive disease, where we really have very toxic therapies and they're not good enough. And so we first started this by thinking about, is there, this business of overdiagnosis and overtreatment, is it real? If it is, there should be actually something that is a threshold that says these are the tick marks on the left are the number of metastatic events. So is there a threshold that we should where we should be thinking, is something not cancer? And this is actually from the original Transbig series where we identified that, in fact, this indolent threshold, we set this threshold and said, okay, let's find a data set. We know, we've looked to see that over time, we've increased the number of low-risk cancers, and we've increased the number of indolent or ultra-low-risk cancers. But can we prove that those, if left alone without any systemic therapy, really have a good outcome for up to 20 years, which is really important in breast cancer? So we were actually really lucky to collaborate with a group in Stockholm, with Linda Lindström and the STO trial colleagues, and find this old trial that was started where women were randomized to endocrine therapy, 2 years of tamoxifen versus not, essentially. And of that, we were able to actually get 45% of the samples from the trial. These are really old samples. And actually, we were able to get 90% of those blocks profiled and get expression profiles to be able to ask, does this indolent signature associate with a very good long-term survival? And here is this data. This hopefully will be published next month. But in fact, those people with the indolent threshold really do have, these are people now without any treatment over 20 years. They were treated surgically, but no systemic therapy. So the question is this a systemic risk? The answer is no, and here it is. Even with just 2 years of tamoxifen, this is just probably a preventive intervention. But this is huge. This means that when you are screened, you could be asked, what kind of cancer do you have? And this may represent up to a third of the cancers that are found. These are postmenopausal, node-negative, low-grade tumors that would give us an opportunity to do less, do simple excision, avoid radiation. So I think that's a really important. Now, the important thing is that indolent tumors are not just low proliferative lesions. On the left, you can see the composition here, that they are, in fact, mostly hormone-positive luminal low proliferative lesions, but only on the right, and on the far left, you see that only a quarter of those that fit that definition really meet this really close criteria. And so now what we're really trying to think about, well, what are these genes? And can this be something that can be used to look at thyroid and prostate, and even in lung cancer, now that we're screening, we're finding these indolent cancers, to avoid overtreatment, because people hear the word cancer and they think, oh my God, I have to do something tomorrow or I'm going to die. Like AML, which is one of the few things that really is a crisis, breast cancer is something where you can be more thoughtful and think about it. So one last comment is that this should also help us redefine the targets for screening. We don't want to find these indolent cancers because early detection is not going to make a difference in the outcome. In fact, it turns out that size is more important once you get these out of the denominator. But even more importantly, we don't want the precursors of these, which we're finding probably at 50,000 a year, over-treating people, doing things that really do can wreck the quality of people's lives. So this is an important goal for the future, and a huge effort to do this work. So how do you start thinking about moving from precision and thinking about moving from debate to wisdom? So in the state of screening, we mostly argue about everything, and we're doing the same thing we did 30 years ago. It's mired in controversy, and it's about when to start, how often to screen, and this problem of overdiagnosis. But in fact, it's an opportunity for improvement. We could build a sustainable infrastructure to address a really critical health issue that's resource intensive, that we spend over $10 billion a year on one single test. And we could do it probably for so much less. And women are caught in the middle, and some are choosing not to screen at all. So what screening really should be is based on advances in risk assessment, biology, more effective at finding these relevant cancers. This probably is a way for us to even be thinking about prevention and make it more cost effective. So that's actually what I like to use this. The University of California president is Janet Napolitano, who when she was Homeland Security Secretary, she brought us PreCheck. This is a great analogy. She told me I had to think bigger. I love that. And I said, okay, I love this PreCheck analogy. So screening should be keyed to risk. And so we don't need to, it's just like treatment should be keyed to risk. We don't want to screen everyone. We want to find the people who should be on the no-fly list and think about them. And by the way, that was a picture in South Dakota, so that's reach, that's scalability and reach for PreCheck. So we developed this trial called Women Informed to Screen, depending on measures of risk, and it's funded by PCORI. We run it through our Athena Breast Health Network. We've come together, we have this network called the Athena Breast Health Network across the 5 UCs, and we've now expanded to Sanford Health in the Midwest. So we have now 150,000 women in this network, and we're really trying to use this as a way to have common ways of collecting information, treating patients, and running large-scale efforts like this. And the design is a pragmatic trial, and we're randomizing patients to either annual screening or personalized screening. And you can choose, if you're not at equipoise and you don't want to be randomized, you can choose which arm you want to go into and be in the observational cohort, because everyone should be in the trial. We should learn from every patient that we see. We've opened the trial. Actually, we have about 1,500 patients of the 100,000 people we're trying to enroll. So far, 66% of people are willing to be randomized. And of the people in the observational cohort, 80% are choosing personalized. One of the things that we are looking at is all the factors that go into risk. We're using as a baseline the breast cancer, the breast screening consortium cohort to put this together. And we actually have, this is the first application of population-based genetics, where we're trying to determine risk screening and prevention recommendations. We have a partnership with Color Genomics. For the price of a mammogram, we can get all of the genes that put people at risk for breast cancer, plus 97 single nucleotide polymorphisms, which I'll show you how we use those in a minute. The model's updated as the study progresses. The idea is to treat based on the current model. not to have something outdated in 5 or 10 years, but to say, well, I'm testing risk-based screening. So as the model changes, we can do that, adding SNPs for African Americans, adding SNPs for Latino women as we go through is really important. So we calculate the polygenic risk score. All of this is done in a completely automated way. We use the Salesforce platform. All of this is integrated. We can actually modify the polygenic risk score is the sum of all of these SNPs, and it actually can move up or down, and I'll show you how that works. We look at the product of the polygenic risk score is based on the product of the likelihood ratios for each one of the individual SNPs. And what's important is that this is the baseline breast cancer surveillance consortium model, and you can see that actually in the low risk, this is the BCSC model alone, and here it is with the SNPs. And you can see what it does is it drives people to that lower risk group in the controls. And so on the low end, it's finding more people with very low risk, and on the high end, it's actually finding more of the people at high risk. And I think this is important because we want to adapt over time. So if you are at risk for an ultra-low risk cancer, or you are not at risk at all for a cancer, we want to be able to find the populations that we don't need to screen where you're going to do more harm than good. And I think that discrimination is not shown by the AUC curve, so you have to really look and see how you're really making a difference in the score, not just on the AUC curve. And here are our buckets for how we assign people to screen. We don't start screening until you have a 5-year risk of 1.3% at 5 years. And for the moderate penetrance mutation carriers, they get annual mammography. And for the really high-risk people, they get annual screening plus MRI. This is a much less use of MRI. Again, we want to do good, not harm, and the false positives are very high. So this is also, you have to take the case to the public. We ask people to share their wisdom. This is our website, the wisdomstudy.org website. And everything can be done now. It's done all automated. I have actually the Wave Analytics output on my phone every morning. I can look and see how the study is accruing. I saw 200 more people on today than yesterday. I was very excited. So our starting bounds our recommendations are all within the acceptable practice standards, but we want to move from there. We want to have a platform for rapid change. We want to learn who is at risk for what kind of cancer, and then tailor screening so it can have the biggest impact. And having a fixed price and a low transaction cost to serve the public so payers can serve their constituents. That last part, I won't say just how much work it is to move that forward, but that's a whole talk on itself. I'm going to finish with the I-SPY trials because this is now thinking about who's at risk for the highest risk cancers. These are the people that we need to make the biggest change for. Screening is not going to affect these kinds of cancers. These are the fastest growing cancers. And again, you have to start treatment earlier in the course of disease when cure is possible, when you have early endpoints and readouts are possible. And clinical care has to change to make it easier and seamlessly to accrue these patients. About 50% of the patients we screen go on these trials when offered, not 5%. And innovation in the standard of care has to be forward, and we have to have a formula and a way to decrease toxicity, and that way we can afford some of the newer drugs that are possible. I think particularly in solid cancers, this article from Andy Grove from 2005 is a real indictment of how we think about things. And he said our knowledge turns are way too slow because we use survival as an endpoint. And as Brian mentioned earlier, I used actually imatinib as my standard because it doesn't really move the needle in blast crisis, but it made a big difference in the accelerated phase. So that's really important. So again, if you think about the standard way we develop drugs, we do phase 2 testing and phase 3 testing in the metastatic setting. You're looking for a signal, and then you would turn to breast cancer, take away your biomarker, which really is the tumor, and then add chemotherapy plus or minus a drug, accrue all your patients, and then do follow-up. There's no way to get that knowledge turned to less than 15 years. Unacceptable. So instead, the minute you have safety data, you have a promising regimen, start in the neoadjuvant setting in the patients at highest risk for early recurrence. Focus on that group of patients and say, plus or minus chemo, does the tumor go away? You find that out in the course of surgery. It's in the course of care, and you could then get accelerated approval. When we started this trial, we didn't have accelerated approval, but we worked with the FDA, got a path forward for accelerated approval, and I'll show you how we're continuing to move that model forward. That would actually take 10 years off the clock. So we're waiting. We're hoping later this year. I-SPY 2, again, is a way to screen these novel agents in combination with standard chemotherapy, plus or minus a novel agent. Our early endpoint is path response, and the graduation threshold is big signal, 85% predicted likelihood of success in a trial. Again, coming in multiple arms in the trial. And, the goal is to test agents where they matter most, rapidly learn to tailor agents, optimize the success of phase 3 trials, put in all this organizational efficiency, which you just saw before. We started this in 2010 with the master protocol, and using this team approach and democratizing access to data, and not giving people credit for writing their own protocol, but giving them for running an arm in the trial and their responsibility. This is the adaptive randomization schema. I don't have time to keep going through it, but if you reach the graduation signal, you come out. If not, you keep accruing patients, and if it reaches futility, it goes out that way. And that allows you to test multiple investigational drugs from several companies under a single protocol. You just saw that. And allows you to have drugs coming in and out seamlessly. The trial never goes down. We actually, from the time I shake hands with a company to say, I want to put the drug in, to the time I get the amendment written, all the contract signed, and getting it into the trials 4 months now. So that's actually the kind of speed that you want, and have it actually accruing in the highest accruing sites. So since 2010, we've actually now tested 13 agents. 10 agents have actually left the drug. 5 of them have graduated and are on their way to getting additional data for approval. And we currently have really exciting combinations, including combinations without chemotherapy in the trial. And this is actually a great platform. And the idea then, us realizing, oh, well, we don't have to have Taxol. Now we can start taking interesting combinations and start dropping things out, is really exciting. And we have 6 more agents and combinations. We've been able to show that, in fact, people can work together, and you'll be happy to know we have a less than 7-day turnaround time for tumor profiling, and we've been doing that since 2010. You have to work with a really robust company that will absolutely commit to that kind of turnaround, because no one has gotten on the trial. We've now screened almost 2,000 patients. We have 1,300 patients that are in the trial, and almost 1,000 patients who have completed surgery, all with complete profiling. So in that sense, biomarkers are used to refine targets. Early recurrence is our early endpoint. If they're hormone positive, they have to be 70 gene high risk to be because we don't want to give chemotherapy to people who don't need it and aren't going to benefit from it, and where early recurrence is not the right endpoint. The assignments for randomization are currently the hormone receptors, HER2 receptors, and the midpoint of the mammoprint, but we have qualifying biomarkers where we're looking at expression, phosphoproteins, and immune complexes. And this is just one example. So in the PARP inhibitor, one of the PARP inhibitors graduated from I-SPY. So we actually have a whole qualifying process. It's very rigorous to find out, is there something better that we can look at? So we found that what bubbled up to the top was this combination of PARP-7 and the mammoprint the highest group, that in that bottom left-hand corner, where there's all the DNA defect abnormalities. And what we found, this is just an example, so when we look at these drugs, these are probability curves of who's going to work. The veliparib-carbo combination, there was a 52% versus a 22%, so big difference, you can see. But when we looked at these individual biomarkers, not so good, but when you combine the biomarkers, we actually now can get to almost 80% response versus 20% response. That's a huge improvement. And it turns out that 40% of the triple-negative breast cancers have that, and that's independent of BRCA status. And actually what's also important is then you can look at it, this drug did not graduate in ER positive, but you go to the ER positive subset, you can actually do the same thing and say, oh, there's about 10% of patients who also have that huge benefit in the ER positive data set. So that's just a taste of what to do. So we have to refine these intermediate endpoints. We have to refine the predictors, the predictors of the intermediate endpoints, drive up efficacy, drive down toxicity, improve the regulatory pathway, and with a relentless focus on rapid learning. We're going to set this big goal. We just got a program project, hopefully approved. We got a score of 21. unless the NCI doesn't get any funding, we should have it. But our goal is to say we're going to have by 2022, that's 5 years, we're going to have pass CR in 90% of patients and 90% or 75 to 95% of patients not having standard chemotherapy. And the way we're going to do that is now we're going to start learning how to tailor within patients. And the idea is, here are just a couple of ideas that we're going to go debate this weekend is, if you don't get a pass CR, if you get a pass CR and you get it early without, in the first regimen, you can go on to surgery. And that would be a path forward. And if you don't get a response, then you can get reassigned to another drug. So everyone can go to the center. And just saying that we're looking at, this may be important in the immune cells, and these are the kinds of things that we're looking at to try and say, so everyone should get a chance. We want every patient to get to PCR. So I think you've heard from Brian and I that the status quo has got to go. We have to have these patient-centered trial approaches that enable a regulatory path forward, trial designs that really allow rapid and adaptive learning, platform trials that promote this approach, and these trials should reflect the way we hope clinical practice will be and how it will evolve, and we have to work hard to develop these affordable and less toxic treatment approaches and integrate the systems of care and research so that everyone's practice is part of the goal of learning. So thanks.
Thank you. Thank you.